microbial growth 2. Recall that microbial growth is divided into two: the open system and the closed system. This is dependent on the availability of nutrients. When there is renewal of nutrients, the system is said to be open. So what are the features of an open system? We have number one, nutrient renewal. Number two, all growth phases are observed. Number three, waste products are removed, and consequently, no accumulation of toxic material. Right. For a closed system, there's no renewal of nutrients, right? That's one. Number two, cells undergo 5 phases only, 5 phases of growth, right? And the next thing is that no waste products. That's number three, no waste product removal. And when there's no waste product removal, the system becomes toxic. That is, there will be accumulation of toxic materials, right? So these features can be combined together in a table as the differences between open system and closed system. So let's quickly go through these differences. Number one, you have nutrient renewal for open system. Two, there's no nutrient renewal for closed system. The same thing for number two, all growth phases are observed. Here, only 5 growth phases are observed, and the 5 growth phases are dependent on the acronym D-cellar, right? From our last video, we talked about D-cellar, right? LA tells us your lag phase, right? E tells you the exponential phase or your log phase, right? Then your S tells you your stationary phase, and the second but last tells you the death phase, and the last one is your decline, the phase of prolonged decline. So these are the 5 phases undergone by a cell in a closed system, right? Now, number 3, waste products removal for open system, no waste product removal for closed system. Number 4, no accumulation of toxic material. Here there is accumulation. And the last one is that this is carried out in a chemostat. It's carried out in a chemostat, while the other, number 5, is carried out on an agar plate or in a test tube. These are the 5 major differences between the open system and the closed system. So quickly, let's look at the environmental factors that can affect or influence microbial growth. Number 1, we have temperature. The next is oxygen. The next is pH, which means power of hydrogen ion concentration. Conc. The next is water. For temperature, it is subdivided into 3. We have the minimum temperature, the optimum, and the maximum. These 3 makes up your temperature range. So for temperature range, the classification is this. For minimum range, we have the cyclophiles, which operates below 20 degrees Celsius, right? The cyclotroph are bacteria that operate around 20 degrees Celsius and above. 20 degrees Celsius. So for optimal range, we have the mesophiles, which operates between 20 degrees Celsius to 45 degrees Celsius, right? Then we have the maximum range. We have the thermophiles and the hyperthermophiles. For thermophiles, they operate between 45 to 70 degrees Celsius, right? For hyperthermophiles, they operate between 70 and above, and any temperature greater than 70. So for explanation's sake, we have the minimum range first. The minimum range is divided into the cyclophiles and the cyclotrophs, right? What are the cyclophiles? The word cyclo means cold and philes means living. This means they are bacterial cells or organisms that live in cold environments. And these are their features. Number 1, they operate below 20 degrees Celsius. And 2, they are restricted to cold region. cold region, right? Examples of bacteria that fall in this category is Bacillus and Staphylococcus, right? So we have the next one being sacrotroph. The word sacro still means cold, and troph means nutrients. In a word, it means bacteria that live in cold nutritional foods, right? Or that are present in cold nutritional food. Now, what are the features of this kinds of bacteria? They operate between the temperature range of 20 degrees Celsius and above. They're responsible for spoilage, right? Example is your Staphylococcus, right? We have the last feature being that they are present in nutritional food. Example, you have your milk, banana, right? We have also tomatoes, right? Right, and so on forth. The next is your maximum range or your optimum range. And your optimum range, you have only the mesophiles, which are responsible for the cause of disease. Example is your streptococci, pneumonia, then staphylococcus, right? The next stage is your maximum range. Here you have the thermophiles and the hyperthermophiles. We've talked about their range earlier, right? But note that the hyperthermophiles are found in deep seas of about 120 degrees Celsius. This class of thermophiles are actually called the extremophiles, right? Extremophiles are subdivided into basophils. They are bacteria that can withstand high pressure, right? We have hyperthermophils or thermophiles, right? They can withstand high temperature, just like we are treating now. Then we have the halophiles that can withstand high salinity level. Does that make sense? All right, the next stage is oxygen requirement. Yeah, oxygen requirement. All right, for oxygen, it's subdivided into aerobic, anaerobic, facultative anaerobic, micro aerophiles, right? The aerotolerant anaerobic. Does that make sense? These are the 5 subdivision for oxygen requirement. So the aerobic are obligate aerobic. Does that make sense? And they require basically oxygen, and without oxygen, they cannot grow. Does that make sense? So example is your gram-positive cocci, right? And your micrococcus. Does that make sense? All right. So we have obligate anaerobic. The word obligate means they do not change their method of living. Does that make sense? They cannot switch to being anaerobic, right? So this does not, they cannot switch to be aerobic, rather, right? They do not require oxygen for their survival. Example is your Clostridium tetani. Your Clostridium tetani is the bacteria responsible for tetanus, right? Then we have facultative anaerobic bacteria. They can survive in the presence of oxygen or without oxygen. Does that make sense? All right, we have the next example of facultative is your yeast. Then we have the next microaerophiles. They require little amount of oxygen, right? Example is your Helicobacter pylori, the one usually found in stomach, right? The 5th one is your aerotolerant, right? Aerotolerant anaerobic. Aerotolerant anaerobic. Does that make sense? They are bacteria that they are basically anaerobic. Does that make sense? But they can tolerate the presence of oxygen for their growth. You get that? So they do, but basically they do not require oxygen for their growth. The one but last stage, we have pH. The pH divides bacteria into 3 stages. We have the neutrophils, we have the acidophiles, and we have the alkalophiles. For neutrophils, they operate between, so this is the pH scale. So they operate between 5 to 8, right? Then we have the acidophiles. They operate below 4, and the next one is your alkalophiles. they operate from 8.5 to 14. This one operates from around 4 or 4.5, sometimes 5 to 1, right? If that is the case, can we quickly treat the next stage, which is the last stage for the study? We have the water requirements. It's divided into two. We have the osmotolerant and we have the halophiles. The osmotolerant, they can be found in water concentration that is 10% below or around 10%. Does that make sense? But for this, they are halophiles. We treated these halophiles under the extremophiles as one of the examples of the extremophiles earlier, right? They are bacterial cells, or they are bacteria that can be found in an environment with greater than 10% sodium chloride concentration. All right, with that, we are through with environmental factors that affect microbial growth. Thank you for watching.